Hello friends. Recently I made a video in which I have told that I will be making videos for the students who are CSIR aspirants or someone who wants to clarify the gate or the ICR net examination. So here there are 3 lecture series which I am introducing here. So this will be a 5 or 10 minutes lecture, so you can get the concepts very clear. If you have any doubts, so then you can always come up with your comments, so then I can clarify that doubt. So the first video, which will be the genome size and the C-value paradox, what does it means. The second one is again a short video which will be what is the relationship between the genome sequence across the species, and here the focus will be on the eukaryotic system. And the third one is a introduction to the NGS technology, what it can do. So let's look into the first video. Now in my previous video I have mentioned, which if you have not come across or your first time watching it, then I will share that link with you as well, or you can visit my channel. Now if you look into the genome size, like a genome size of a prokaryotes, when we talk about the prokaryotes, when we know that this we are referring to either the bacteria or we are referring to the archaea, right? So they have comparatively a not a well defined cell structures, and they lacks the nuclear membrane. So they do have a very small genome size, and the number of the genes is also comparatively small. But as you are going up from a lower eukaryotes to fungi to the protists to the animals and to the plant systems, then you find that there the genome size is increasing. We will see a table in a minute. So what the statement which is defining here is that the genome size correlates with the cell size, right? The division rate, like how much it is dividing, as well as it is depending upon the metabolism, the development rate, or the body size. So there is a pretty much acceptable, you can say, the notion that the genome size should be depend upon that. Now we know, as I mentioned also, that eukaryotes have a well-defined nucleus. They have genome which is packed by the nucleosomes, by histone proteins, then further packaging, folding to make a chromatin, and from then chromatin then you see the chromosomes in the metaphase stage. However, there are few exceptions in the prokaryotic systems, and I'm telling you so that these types of questions, if it is asked in CSIR or UGC or GATE or national level examination, then you can able to attempt. And what are those exceptions? There are exceptions in prokaryotic systems, like some bacteria also consist of a linear chromosome. Now usually the bacteria is we know that is a circular, they're minuscule in size, and they have it is not a linear chromosomes which is present. In fact, the chromosome terminology is a misnomer which is also being used, but let's for a definition sakes we are saying it is chromosome. Then they also have a linear chromosome. An example is streptomyces. The another exceptions which you find that there are multiple chromosome size plasmids which is present, means a huge plasmids, and one of the examples is the Vibrio cholerae, which causes the cholera, is an example. Besides this, you also find that there are certain hallmarks which you find only in the eukaryotic systems, like in terms of your introns or the transposons, specifically like the insertion sequences is also present in case of the prokaryotic system. Now if you compare the genome, which in the introduction I was trying to mention that if you look into the genome of the bacteria or the archaea, they are from few kilobases to a 15 megabase, which has been reported so far. However, if you consider a eukaryotic system, the smallest genome size which is been known for the encephalotozone intestinalis, which is one of a protist, it's only a 2.3 megabase pair, very comparatively small for all eukaryotes into consideration. Even like for, if you see the yeast, the Saccharomyces cerevisiae, it is having a genome sequence of around 12 megabase pair, right? So that is, and if you compare, it is very comparatively small. And then if you look into a plant system like Paris japonica, it is having a 150 million bases. That's a huge size, and that's probably explain the complexity of the organism which we are looking in the previous slide. Now, if you see in this table, which has shown that the Saccharomyces cerevisiae, which is a genome size of 12 million bases, it is the gene coding is 6,000. If you go to a other increasing order, okay, or say a few examples like malarial parasite has been 23, the plasmodium is having 23 million, and the genes is roughly 5,000. Now if you compare this and this here, even in this diagram itself, you find that even with a genome size of like approximately twice, the gene number is not increasing. Okay, that's what you observe. If you go with the Drosophila melanogaster, like one of the model organism very popular in molecular biology and genetics, it is having a 170 million bases. Okay, if compared to the Saccharomyces cerevisiae, how many genes which is been present, which is roughly you can say more than 15 times the genome size, but there is the gene numbers which we find is only 14,000. If you compare that with a human being, which is roughly we see that is a 3 billion bases, the human genomes, there are genes number which is present is 20,000 to 25,000. The point which I am trying to make here is that with the complexity of the organism, the genome size increases, but we find there is a disparity, right? There is a disparity which has been found in terms of coding sequences or the protein coding genes which has been present. This terminology is actually defined as C-value paradox. This means the C, which in literature so many definitions are there, but the acceptable one, it is the constant. The C stands for the constant, and it refers to the DNA or the genome content. Okay, so that is the C-value paradox, which suggests that with the genome which is having a large genome, it is not encoding the exact protein coding sequences, and we found that there is, based on the size differences or a cell or as an organism whole, we do not find any correlations between the gene numbers as well. Like for example, if you take an example of a human cell, and if you take an example of a yeast cell, yeast which is a unicellular organism, there are 12,000 genes, but in case of humans, which is roughly 22,000 genes. It doesn't make sense, like in looking into the complexity of the organism, and that's what which is called as a C-value paradox. So if you look into the definition of C-value, it is termed as it is a total amount of the DNA in the genome, and it is expected, like more the complex the organism, the more DNA is to run it, but in fact we do find that this relationship is not existing, right? It appears to be it's making a sense, but we find that this is not so, and that's why it is called as a C-value paradox. Now this leads to some interesting and important questions as well. And what are those questions? Like, what are the, if we compare let's say example of a barley genome, which is roughly around like say 38 megabases. Now this barley genome, there are very few genes which is coding. Or let's say take example of a human being. Now if even in the human beings which we are saying that it is it is 38,000 barley, 38,000 megabases, and in case of human, which is roughly like 3,000 megabases. These the gene numbers is roughly 22,000. So what are the other sequences which is present in these organisms? This will be dealt in my video number 2. So we will discuss that, but it raises the important questions that what are these sequences. Obviously these sequences which we say is the repetitive DNA sequences which can play important role in the regulation. So but still it is not very clear, and the reasons for this is the amount of the genome sequences which is available. There are roughly so far 15,000 or the organisms so far the genome has been sequenced. There are still more genome has to be sequenced, then we can get a little bit better idea. So that's what the C-value paradox is coming into picture. What is the evolutionary time frames of these elements? How they've been accumulated? Is it there is a gene duplication, but why it has been still there, or it is disappear from certain regions? So why that is so? Is there is any kind of a positivity or negativity related with its coding and the non-coding and leads to the organism biology? These are very important questions which one has to address. So we will see in our lecture video number 2, based on this, that is the molecular biology aspects, that is there any kind of relationship we find. So in summary, I would like to say that we find that with the complexity of the organisms, the C value is increasing, but we find that there is no correlation between the number of the coding genes. So that's the C value paradox, and we do find that there are several unanswered questions which needs to be addressed. Now if you look into the repetitive DNA sequences, so these could be highly repetitive or a middle repetitive sequences. Like highly repetitive is satellite DNA. If you look into the middle repetitive sequence, a repetitive sequence means one of the sequences coming again and again. Okay, so that could be a tandem repeat, or it could be a retrotransposons. Like one of the famous which we know, the lines and the signs, which roughly consist of like 45% of the human genomes. There are mini satellites, there are microsatellites, and so on. So what is the function of these repetitive or non-coding sequences is still is a question of exploration and research. With this I will stop here. If you like these things and found it to be informative, you please subscribe to the channel, share with your friends, and at least give a thumbs up for this. Thank you very much.